Hello, everyone. My name is Mina Napavi, and I am a program manager with the Education Core with the UAB CounterACT Research Center of Excellence in Arsenicals. Welcome to today's Grand Rounds seminar. For those of you not familiar with our center, it is led by Dr. Mohammad Athar, and the goal of the center is to develop mechanism-based, highly effective medical countermeasures against arsenical chemical warfare agents. Our next Grand Rounds will be on September 21st at 12 p.m. noon, when Dr. Ziad Kazi presents on the medical and public health considerations of the Beirut ammonium nitrate explosion. For those of you involved with the NIH CounterACT program, please remember to save the date for the next NIH CounterACT Symposium, which will be held on December 6th through 8th of this year. The UAB Research Center of Excellence in Arsenicals will be hosting NIH's annual CounterACT Research Symposium in New Orleans, Louisiana. For more information on the center or its activities, please visit our website at uab.edu/arsenicals or send us an email at as33@uab.edu. On our website, you will find links to recordings of past Grand Round presentations and our podcast. So it is an honor for me to present Dr. Mohammad Athar as our guest speaker and director of the UAB Research Center of Excellence in Arsenicals. Dr. Athar is also a distinguished professor and vice chairperson of dermatology in the UAB School of Medicine, where he is the inaugural holder of the Eric W. Baum, MD, Endowed Professorship in Dermatology. Dr. Athar has received multiple awards, including the 2019 Dean's Excellence Award in Research. And over the past 3 years, that's 2018, 19, and 2020, the Blue Ridge Institute has ranked Dr. Athar as the #1 principal investigator in dermatology. In addition to his many accomplishments, Dr. Athar has mentored approximately 34 postdoctoral and clinical fellows. He has authored or co-authored over 240 publications, has completed 60 reviews of book chapters, commentaries, perspectives, and editorials. He has 2 patents and has authored 1 book and co-authored 3 others. His background is in skin biochemistry, signal transduction, toxicology, and carcinogenesis. Currently, his laboratory is focusing on animal model development for skin cancer and skin inflammation or injury, in which his work is focused on the identification of molecular pathogenesis of the disease, molecular driver pathways, and their molecular mechanisms, employing several modern tools and techniques, including bioinformatics approaches. So welcome, Dr. Athar. We're thrilled to have you speak today.
Thank you, Mina, for a very gracious introduction. I'm delighted to present my work to the audience today, and my topic is on the molecular underlying mechanisms in cutaneous chemical burns caused by arsenic exposure. Although our center is working on the injury produced in multiple organs, but today's presentation is only related to skin, and rest of the presentation will be given by other investigators so that everyone knows what is going on in our center. Everybody knows, but for those who do not know, arsenicals are highly toxic chemicals which were developed during World War I and World War II, and their stockpiles still exist in some places, and I will show you a map where they are. And these chemicals are highly toxic and cause deleterious effects, which one may not presume that such small tiny chemicals, how can they induce such damage? So Organization for the Prohibition of Chemical Weapons, they completely blocked the stockpiles of these chemicals or their generation anywhere in the world. They monitor all over the world the stockpiles of these chemicals, their synthesis, and so on and so forth. The chemicals that I will be presenting, they cause inflammation and pain in the skin and in other organs when they are exposed, if you see their structures, you will find that these chemicals are very tiny arsenic containing chemicals, and just a small change in the structure can change the biomedical properties or damaging properties of these chemicals. For example, the most devastating chemical lewisite is a vesicant. When it is exposed to skin, it causes blistering and inflammation and pain within few minutes. And then if you see the other chemicals where arsenic is attached to a phenyl group or a chloro, it is a strong vomiting agent, whereas cyano compound with similar structure is sneezing or lacrimating or coughing agent. So these agents, they change their properties as their chemical structure is altered. Now, we also have developed a less potent and less toxic small molecule to show that it can produce similar effects at higher doses. But this chemical can be used in the laboratory, whereas warfare grade chemicals can be used only in authorized companies, and we use the company known as MRI Global, which is located in Kansas City, Kansas. So this is the map of arsenic and arsenic containing chemicals. You can see that arsenic is also present in underground water, and environmental exposure to arsenical occurs. That is not the part of this talk, so I will not discuss that. But these other areas are marked where in the ocean there is a dump of these Second World War ammunition, and the ships were sank in the ocean, and you can see that these are all over there. Also, remaining portion, there is a big burial of these chemicals in the US and in Asia, as well as in Europe. So this map demonstrates that where these chemicals exist and their exposure can occur accidentally, or because they are so simple, they can be synthesized with ill intention, and they can be used against public. So why we are so concerned? Because in recent past, there are a lot of accidental exposures that have occurred during the construction, and laborers were exposed, or in China recently, sulfur mustard and lewisite containing drum was exploded accidentally, and about 18 to 20 deaths occurred and a lot of other people injured. Similarly, we have a small leak of chemical in Spring Valley in Washington, D.C., where accidental exposure to lewisite occurred during the construction, and that led to hospitalization of many people, although no casualties were reported. Similarly, the degradation product of Clark 1 and Clark 2, which is also, as I showed you, lacrimating agent and a warfare chemical. This is diphenyl arsenic acid, and this poisoning occurred in Japan because of the leakage in underground water table, and that led to neurological syndrome in exposed populations. They are still there, and they are being monitored with various techniques. So all these accidents tell us that we should know more about these chemicals and be prepared for any emergency that may occur, either accidentally or through the nefarious purpose of using chemicals against human population. So our purpose is to develop a suitable model for understanding the pathobiology or pathophysiology of these chemicals. And we developed murine and porcine models that we will show against these vesicants or irritating chemicals. And then we try to uncover the molecular mechanism by which they cause these pathobiological or pathophysiological changes. And then based on those mechanisms, we try to develop some medical countermeasures or antidotes. As you see here, this is the mouse skin data showing that leucite and other chemicals, how they are different. They were exposed on this mouse skin topically, and they were exposed in molar equivalent doses, so that we can see that at a constant dose level how different they produce effect. So you see, in case of leucite, there is a time-dependent injury that could be visualized up to day 168, and there is some recovery also at this point. But other chemicals, they induce much less injury, particularly this last chemical, difluorochloroarsine, produces much less injury on the skin, although it produces certain changes in the skin. Similarly, we expose these chemicals to porcine skin, and you can see that in the porcine skin, these chemicals cause blisters similar to those which are induced in humans. And these blisters associated skin is associated with huge inflammatory response and tissue damaging response. So I will show you in the following slides the mechanism of each of these action. So for example, what happens when leucite exposure occurs? Leucite exposure leads to disruption of basement membrane, and you can see in mouse skin that there is a progressive damage to basement membrane, leading to the formation of separation of epidermis to dermis and formation of a micro blister, followed by a big blister formation. So this is just not the basement membrane, but also tight and adherence junctions are also disrupted. These junctions contain specific proteins. For example, for tight junctions, we have monitored ZO1, ZO2, and for adherence junction, we monitored N-cadherin. So in next slide, you see that both in murine skin as well as in porcine skin, you see disruption of alpha-ketanine and ZO in a dose and time dependent manner. And here in this slide, although I have not shown all the data, but this data shows that the disruption occurs progressively, and the molecular mechanism involves the dissociation of a protein which is known as YAP, which is a hyposignaling protein, and that when dissociated from the junctions leads to degradation of other complex proteins like alpha-ketanine or ZO1, leading to the disruption of the junction. So this occurs both in murine skin and porcine skin. So in this regard, both murine and porcine skin cause these damages through the analogous mechanism. And if you see progressive damage, you see that damage starts continues to progress, and at later time point you see induction of cell death through the mechanism known as apoptosis. And in apoptosis, these blue cells are skin keratinocytes, and here is a hair follicle. So you see the staining in green. In green, the apoptosis positive cells, which which are progressing for the death. And this mechanism is also common both in mice and in porcine skin. And all the chemicals, they induce apoptosis, although the potential to induce apoptosis is different by different chemicals, Lewisite being most potent compared to DPCA or DPCYA, which are less potent in inducing apoptosis and cell death. As time progresses, the cell death mechanism also changes, leading to the activation of a pathway known as necroptosis. This necroptosis pathway is activated by the kinases, which are known as RIP kinases, and they trigger another kinase, MLKL, which is also a biomarker for the induction of necroptosis. If you see the mechanistic difference between necroptosis and apoptosis, apoptosis is mediated by the activation of P53 signaling. P53 is a tumor suppressor gene, but it also regulates apoptosis by activating executive proteins for the induction of programmed cell death. So the mechanism for the progression of damage are distinct, and they are time dependent. They switch from one pathway to another pathway. And if you see that we have defined time dependent dynamics of the damage, as well as dose dependent dynamics of the damage. Here is the data for Lewisite, where you see that how necroptosis occurs, leading to massive tissue damage, and you see necrosed skin in the bottom in H&E staining over here. And this is the biomarker, as I told you, is overexpressed all over in keratinocytes and in dermis following exposure to Lewisite. This is associated with RIP kinase 1 as well as RIP kinase 3 activation. So then another response to arsenical exposures is the induction of inflammation. There is robust inflammation induction, and we identified that both macrophage and neutrophils are involved in the induction of inflammation signaling. They infiltrate at the damaged site, and you can see by MPO staining, this is neutrophils, whereas F480 represent macrophage. And as far as kinetics of accumulation or infiltration of these cells is concerned, this is different with different chemicals, and as well as this is also dose dependent. And nonetheless, these cells, they accumulate in a big way in both porcine as well as murine skin analogously. However, as shown here, like CD3 staining or some other stainings, there are also additional inflammatory cells like T cells are also accumulated. And this is the data with Lewisite and with other arsenicals that you see that not only neutrophils or macrophage or monocytes are involved, but also other cells like T cells are also involved in the inflammatory response. So what are the mediators of inflammation? And here we saw three specific cytokines and prostaglandins are involved in the inflammatory response in all these chemicals. They are IL-1 beta, IL-6, and prostaglandin E2. Prostaglandin E2 is particularly important in context to skin inflammation, but also in other organs, it is important. And you see that all these cytokines are induced multifold following exposure to these chemicals. So this was our biased assessment. So we also looked into unbiased cytokine and chemokine profile of these chemicals. And you can see that there is a dynamics of cytokine production at 6, 24, and 72 hours. Different cytokines, they are produced in different quantities, and they may be related to progression of injury as well as repair process. Similar changes occur in porcine skin with different chemicals. So we have fully characterized here both tissue damage and inflammatory response, and we wanted to check the mechanism of inflammation induction as we have done for tissue damage induction, that early induction is p53 dependent apoptosis, whereas late tissue damage is dependent on necroptosis. However, in case of inflammation, this is very complex phenomena, and the mechanism involves through different pathways. And one of the key pathways that we recognized was dependent on nucleic acid release. And in this case, we found that pathways which are regulated by double-stranded DNA as well as double-stranded RNA are regulated. So here is a cartoon depicting the induction of C-gas sting pathway, which is activated by double-stranded DNA. And you see that in case of leucite and POW, this data is for POW, and this here is for leucite. Both at mRNA and protein level, there is a significant induction in the pathway, including the TBK kinase, which is downstream of induction of C-gas and sting that leads to activation of NF-kappa B and IRF3, followed by production of interferon type 1. So if you see the other pathway, which is sensed by double-stranded RNA, is here. This is regulated by a protein known as ADAR1. ADAR2 is also involved in this, but ADAR1 plays a major role over here. So we show that ADAR1 is activated both by leucite as well as our chemical POW, and the induction leads to the activation of this pathway that leads to activation of EIF2 alpha and production of interferon type 1. So we confirmed that in the skin, we see high levels of interferon alpha by POW as well as by leucite. So we believe that these two pathways are underpinning of inflammatory response. And also we studied the mechanism by which, there is a production of robust production of cytokines in this way. So here is a pathway that depicts and correlates with the dynamics of damage. We found that in early hours, damage is not as much as in late hours. And one of the pathways that is induced by chemicals is known as stress granule formation. What stress granule does, that they accumulate proteins and mRNA that are responsible for causing cytotoxic response. So early induction of these stress granules lead to protection of tissue against the chemical injury. And we found that this pathway is activated by the exposure of arsenicals, and that also leads to activation of EIF2 alpha, and that occurs through the through its phosphorylation. So I will get back to you because this is an important molecule that regulates protein translation under stress. So I will show you some of the data related to that, but stress granule formation occurs early on, and that is perhaps responsible for the protection of injury at early point. But injury is so huge that these stress granules are not able to control the injury, and therefore they disassemble very quickly. And I show you the dynamics of formation of stress granule at early time point at early doses, where this assembly occurs at up to 3 hours of exposure to POW, and thereafter from 6 to 12 hours disassembly occurs, and that leads to the release of cytotoxic proteins and mRNA. Now, while stress granules are assembled, what happens that they also associate with RNA binding proteins, so that whatever RNA or proteins are assembled, they are degraded in the stress granules. And here is a protein known as roquine is targeted to stress granule to degrade various inflammatory protein, inflammatory RNA for their degradation. At the same time, we also found that there is a similar protein which is known as roquine, but this is targeted to ribosome. So we found that both of these proteins are induced following activation of tissue damage in a time-dependent manner. And here you see a correlation of degradation of roquine and regnase in a time-dependent manner, and induction of IL-1 beta in the tissue. So because these are the RNA degrading proteins, so their deactivation is necessary for the induction of inflammatory response. In order to confirm that these proteins are involved in the arsenicals induced inflammation, we did a siRNA-based experiment in which we knocked down the expression of roquine and regnase 1. So you can see that this is the power induction without knocking down in the control cells. This is IL-6, this is IL-1 beta, and this is COX-2. COX-2 produces prostaglandin 2. So we have monitored these 3 molecules kinetics of induction following knockdown of roquine as well as regnase. So in a scrambled cell, you see that induction of IL-6 is about 40-fold increased following roquine knockdown, and similarly 30-fold induction occurs following the knockdown of regnase. So as I showed you the data that these 2 proteins are degraded in a time-dependent manner, and that causes the activation of cytokine production. Similar responses you can see with IL-1 beta and COX-2. COX-2 is more robust, and you can see 60-fold or even 80-fold induction at certain doses of POW. So based on that, we wanted to understand that what is going on mechanistically during the progression of chemical burns after arsenical exposure. So we ran a transcription reporter assay to find out what are all transcription factors which are involved. And in this case, we found that all the transcription factors from top heat shock protein, response protein, regulating transcription factor, till here you see more than 2-fold induction. This is highly significant, and they are all related to the pathway which is induced by stress. So what is this pathway which is induced by stress? Is stress on ER, endoplasmic reticulum, leads to the impairment in the proper folding of the proteins at ER, lumen. And that leads to either a majority of proteins to be lined up for waiting for proper folding, or if in a timely manner ER stress is not relieved, these proteins are unfolded or misfolded, and that leads to activation of a pathway which is known as unfolded protein response signaling pathway. Now, the purpose of this pathway is to generate in situ a number of chaperone proteins that can bind to polypeptides which are waiting for proper folding and block its degradation. However, if it doesn't appear in a timely manner, they also lead to inflammatory response and tissue damaging response. Yeah, one of the pathway which is very important in this regard is ATF4 and CHOP pathway, and then the other pathway which is important is EIF2 alpha phosphorylation dependent pathway, which blocks about 30% of all the proteins being transcribed at that time. So if you look into this pathway, what are our options for the therapeutics? We found that there are certain FDA approved agents that could be used to test whether those chemicals afford protection against these pathways. So we used phenyl butyric acid as a chemical chaperone that acts similar to chaperone proteins and blocks the degradation of or misfolding of various proteins which are not folded properly. And then there is an antioxidant, N-acetylcysteine, which blocks the production of reactive oxygen species. So see here, this shows the data how arsenical induced reactive oxygen species can lead to activation of UPR signaling. And here is our data both in porcine as well as in murine skin. You see induction of ATF4 and CHOP pathway in the skin keratinocytes. This is both in we have done these studies both in with POW and other arsenicals, but here is the data for arsenical leucite as well as for other chemicals both in porcine and murine skin. So this pathway is tremendously activated, and when we ran RNA-seq, we found that all the genes regulated by these pathways are activated here. So here is the data to show you that leucite or other arsenicals induced ROS, if blocked by NAC, what it does, so we used both PBA as well as NAC and its combination, and you see that this combination both in the pig and in murine skin leads to diminution in the production of ROS. And then when we applied these chemicals on the mouse skin, you see that this is the leucite skin, and this is for PBA, and you see significant reduction in the tissue degradation. With NAC, it is much more impressive compared to 4-phenyl butyric acid, and in combination, we almost attenuated the damage. And if you see that whether it is dependent on UPR signaling, you see reduction in ATF4 and CHOP by the combination almost 80 to 90%. So this is the quantitation of the data, and this is we ran RNA-seq and ChIP-seq to show you that how a reversal of leucite-induced mRNA is attenuated by 4-PBA or NAC treatment. So in next slide, we show the damage that is controlled by this skin, and there is a decrease in inflammation infiltration of leukocytes in the skin treated with 4-PBA and NSE. And then if you see the translation, you see with POW, this study was done to study the translation of nascent proteins, and puromycin, it binds with the nascent proteins produced transiently in response to any stimulus. Now, when we treated with POW, you see complete diminution of this signal shown in red here in the control. This signal is very good, but with POW treatment, this signal disappears. But when we treat with NSE or 4-PBA, this signal come back, and with a combination, this is much more more close to the control nascent cell nascent protein translation. So it is clear that the translation, protein translation, is restored by the treatment with these chemicals, and that is dependent with the disappearance of phosphorylated EIF2 translation factor. So there is a small molecule known as Isolip that blocks the global stress response. We use this chemical, and this chemical blocks EIF2 alpha phosphorylation significantly. And you can see here that both tissue damage as well as inflammation is completely blocked by the Isolip. This also leads to diminution in the EIF2 phosphorylation at the stress granule, and recovery of roquine and recnase following the treatment. And this is here shown certain dot plots where we showed that how it leads to diminution in the cytokine production. So with this, what we believe is that these warfare vesicant induce severe tissue injury by dysregulating multiple homeostasis control, and that occurs as transcriptional level, post-transcriptional level by changing the binding RNA binding proteins, and also translational level. And based on that, we developed therapy which is known as chaperone plus antioxidant therapy. And we believe that 4-PBA being already FDA approved for certain purpose, we can relabel it and repurpose it for the treatment of skin injury. And NSE being antioxidant can be treated as GRAS. GRAS are the chemicals which FDA considers as generally, GRAS stands for generally safe chemicals. So those chemicals can be, if they are combined, that's not known as combinatorial treatment, although they provide an advantage to the formulation. So our formulation is being done based on that conception, and we are making a formulation for chaperone therapy. Now, having said that, then we also looked into other aspects of these inflammation. And one of the known aspects of inflammation is infection, which is caused by bacteria or viruses. And here we compared the infection caused by bacteria. One of the component is LPS, and LPS induced inflammation in sepsis. We took all the data to compare it with the inflammation that is caused by chemicals. And in this section, you see that both in pig and in mouse, there are comparable size of genes which are common in both the inflammation types, both caused by bacteria as well as during sepsis as well as by our chemicals. And the purpose was to develop a hypothesis that how the two chemicals and how the two different processes induce similar kind of inflammatory signaling that we observe. So we found that the inflammation regulated by bacteria or viruses involves a protein which epigenetically regulates histone opening, is known as Brd4, and therefore we hypothesized that these chemicals, they dysregulate Brd4 and induce injury, including inflammation. So while we were doing these studies, recently we found that Brd4 has become very important, and its inhibitors are being developed for a large number of diseases, including COVID recently, but also inflammatory diseases or cancers or some other. But the target being, the main target being either proliferation or inflammation. So what does Brd4 does? This is the normal chromatin structure, which is highly packed. And when there is an acetylation on the histone tails, this structure is altered and it starts opening as a result of acetylation. And these acetylation marks are produced. Now, this acetylation mark is read by a protein known as Brd4. There are other BRDs, bromodomain bed proteins, which are also there, but they are less studied. However, the domains are common, which regulate inflammation and proliferation in Brd4 as well as other bed bromodomain proteins, namely Brd2, Brd3. So when they are opened, they lead to the expression of inflammatory genes, and as a result, you see instant inflammation. Shown here is the data for leucite exposure, and you see that this is a global acetylation assay in which we found that leucite exposure in the skin leads to activation of pathways that leads to hyperacetylation of proteins. Now, among them, we also typed Brd4, and we see that Brd4 is also induced. When we looked into the Brd4 regulated mechanisms in the skin, we found that histone marks at histone H3, histone 3, and histone 4 are hyperacetylated. So indeed, that shows that the Brd4 related response is activated following exposure to leucite, and these are all the genes which are regulated through Brd4 in an RNA-seq heat map, and then we confirmed these Brd4 target genes using our RT-PCR platforms. So we are sure that this epigenetic mechanism is involved in arsenicals exposure, and you look here for other chemicals, similar response occurs. So I'm quickly changing this to other slides showing in porcine skin that this is also common. So we are sure that inflammation induced by these chemicals, both in murine skin and porcine skin, occurs through common mechanisms, and both are good models that we have developed to study and develop medical countermeasures against arsenicals toxicity. Now, these are the drugs which are currently in clinical trials with Brd4 inhibition, and one of the drugs which is in phase 3 trial for myelofibrosis is CPI 06610. So because this has been very well studied, we selected this drug to see the effect of this drug on arsenical-induced alterations in the skin. And you can see that we applied POW followed by application of CPI 5 to 10 minutes after application of POW, and then harvested skin at 6 and 24 hours. Here is the data. You see a significant inhibition in the acetylation pattern, Brd4 expression, as well as histone mark H3 K18 acetylation and histone 4 5AC. Also shown here is the decrease in the transcription response in the next slide, and these are the transcription marks with inflammation or tissue damage, one representing each. As for example, NLRP3 is an inflammatory marker, whereas others are tissue damaging or tissue repair markers. They are positively regulated by the treatment with CPI. If you see the gross protection against the inflammation by CPI, you see significant attenuation of the response. So until here, we identified several compounds. One is with epigenetics mechanism, and then the others with other protein regulating mechanisms that regulate the damage. We are also working with SR to develop certain compounds, and these compounds are unique in the sense that they block two targets together. So one target is RIP kinase that you saw that leads to the activation of necroptosis or necrosis in the tissue, and then the other epigenetic target Brd4. So they developed for us a very novel compound, which is a dual inhibitor of Brd4 and RIP kinase, and we tested it here in the similar protocol that we tried with the other clinically relevant compound. So we treated mouse with POW, and then after 5 to 10 minutes, we treated with SR's Brd4 and RIP kinase dual inhibitor, and we collected the tissue at 6 and 24 hours. See here, you see a significant diminution in the inflammatory and tissue damaging response. And this is dependent both on RIP kinase signaling. You see decrease in RIP kinase signaling as well as Brd4 signaling. So we believe that this compound is going to be very effective and very novel compound. We are very excited about it, and we are thankful to SR to develop this compound with our thinking that dual inhibitor can be very effective. This is just to confirm in different time intervals that and different doses that this compound remains effective. Here is the data with at mRNA level. We confirmed with the pathways downstream of Brd4 that how this SR compound reacts and reverts or attenuates signaling. So you can see that we see significant decrease in the genes which are induced by POW, which is related to either inflammation or tissue damage. And so we believe that we have a novel agent also in hand that could be very useful for the diminution of injury. In this way, we developed a pipeline of agents, and you see how many agents we have tried, and we have all the data, positive data for their development. The first two agents, NSC and 4-phenyl butyric acid, are under the advanced stage of novel formulation development, and they are going to go, I think, for the treatment, as well as we will have IP rights on the novel formulation to use it. Their toxicity is known, everything is very well known. We have clearly defined the mechanism. So this agent is going to be forward, but we also have data for other similar agents with different targets showing over here. And in the first part of the slide, these are the 15 compounds that we have tested, and we have all the data, both toxicity as well as clinical data, to show that they are effective. Indeed, our selection is first for this combination. And then we also have agents which are in pipeline. Some of them are with C-gas sting pathway, or other pathways like other targets, for example, Adar inhibitors, or the downstream protein TBK inhibitors, and that are also in progress, and we are working with those agents. So in summary, we have created a big pipeline of the agents that could be very effective. We have very good model, both urine and porcine, to study these and similar chemicals and understand the mechanism of their pathogenesis, that these models recapitulate the human pathogenesis, which is known so far. Whatever we found, that could be very faithfully recapitulated. And then we discovered complex mechanistic underpinning and master regulators of injury. We have created a pipeline of medical countermeasures or antidote for arsenical-induced injury. So with this, I will thank MRI Global for their help, as well as with my big team who has been very powerful for generating all the data. We have in the lab, we have large number of people, but Ritesh and Mahendra have been very helpful in doing some of the experiments. Zipping is very efficient in generating a pipeline of mice that we use, and also arranging transport of animals to MRI Global and back from them. We have a team of people who are doing work with lungs, and Anupam has been working with kidney, but he has been very helpful in orchestrating this U54 or UO1 grants. He's very helpful. Our drug development team at SR is highly competent, and Veena is associated with delayed lung injury. She is a highly competent physician scientist who has been very helpful to me in many ways. And finally, this work would have not been possible if NIH CounterACT program has not supported me significantly. They are continuously supporting me with big grants and big money. Also, they are very excited about my work, and they always appreciate, and they look into newer data that I generate. So they discuss with me a lot of things. So I'm highly thankful to them.
Thank you, Dr. Athar. We do have one question and a comment. It's from Dr. Ziad Kazi. He wrote, "Thank you for the excellent presentation. Are the potential drugs that you're developing effective after the exposure to the arsenical, or are they prophylactic? And if effective post-exposure, is there a time limit after which they lose effectiveness?"
We are optimizing the exposure. Initial studies were to understand the molecular pathogenesis, followed by post-exposure to chemicals. And this exposure was up to 30 minutes we have tested. But the studies which are progressing now, we are testing these antidotes up to 2 hours after exposure to arsenicals. This is what we are doing. Our formulation is very effective. We have developed a foam formulation, and the data, I have not shown all that data, because of the time limits. Foam formulation is very effective. What it does, that when you apply foam formulation and remove it within 5 minutes, it takes out all the unabsorbed arsenicals from the surface of the skin. And then you reapply that formulation second time after 5 minutes, then it delivers the drug also. So it cures as well as it cleans the skin surface. So that formulation, we are very excited about it, and I think is going to be a very novel formulation which has both cleansing as well as drug delivery effects.
Another question from Dr. Kazi. He said, thank you, Dr. Athar, for this pioneering work. Do you predict similarities with sulfur mustard mechanisms of skin toxicity?
Some are similar, but we have done only cursory studies on the mechanisms. Of course, we see a lot of similarities between we usually did nitrogen mustard experiments. We have not done sulfur mustard as yet, but in nitrogen mustard experiments, we see both activation of UPR signaling as well as activation of Brd4 dependent chromatin remodeling.
Wonderful. Well, we are at the top of the hour. So I want to thank you, Dr. Athar, so much for presenting. Thank you to all of our attendees. Like I mentioned before, this recording will be available on our website in the next few days. Again, that's uab.edu/arsenicals. And we look forward to seeing you at our next Grand Rounds, which will be September 21st.